Today's lecture is on the hypothalamic pituitary thyroid axis. What I'm going to do first is to take you to a couple of sites through links on our WebCT homepage and related links through that homepage, as you'll see, to the sources for you to read details on the HPT axis that actually supplement tremendously the content of the PowerPoint presentation that I'll be providing you with today. There's a wealth of information from those resources, as you'll see when I briefly go through them on promoter regions for TRH and TSH and other details of relevant molecular information consistent with the studies that we have initiated in the introductory part of this course and through the methods evaluations. So let's go on over to the WebCT site and I'll show you the the links that I'm talking about. Of course, Endotext is an extremely important resource through the remainder of the semester. And I'm going to take you to the chapter 4 of the Endotext here. It is the normal physiology of the hypothalamic pituitary thyroidal system in relation to the neural system and endocrine glands. I'll just scroll down on this and show you some of the interesting aspects: the regulation of thyroid stimulating hormone synthesis and secretion, the molecular biology and biochemistry. The TSH hormone is discussed first, the alpha and beta complexes, as we will see, the molecule itself, regulation of TSH synthesis and secretion, effects of the thyroid hormone on TSH synthesis. So here's information on the promoter region of the human TSH beta subunit, showing the TREs, the thyroid hormone regulatory or response elements. So those are not in the PowerPoint lecture, but you want to be sure to understand it from this source. In addition, some other regulatory regions on the human TSH beta gene, the positive regulatory regions, the PIT1-like binding sites, CJUN AP1-like binding sites, and the T3 receptor binding sites, or the TRE that was mentioned above. As we continue through with this, there's information on thyroid hormone effects on the release of TSH. So some graphs with the figure legend describing those. This is nuclear T3 nanograms per milligrams of protein graphed against hours, as well as plasma T3 nanograms per ml and plasma TSH percent baseline. So this is a very integrative graph that will be very interesting for you to interpret and evaluate. Here are the basic elements in the regulation of the thyroid function. Our hormone, gland 1, gland 2, gland 3, hormone 1, hormone 2, and hormone 3 model, of course. Another graph showing serum T3, T4, and TSH concentrations in rats receiving low iodine diet with or without potassium iodide supplement in the drinking water. So there should be some integrative biology that you can draw conclusions on or from within that graph. The role of TRH in the TSH secretion. We'll see a bit of that. Again, there's the TRH. The 5' flanking sequence of the human pre-pro TRH gene, showing some of the regulatory elements there. The CRE is underlined. One sequence has overlapping TRE and CRE, so interesting to think what might be going on there. And the GRE is also present on the TRH gene receptor. So the GRE, of course, is the glucocorticoid response element. The TRH interaction with the pituitary thyrotropes and with thyroid hormone. So that will be very important. So again, here's the TSH beta gene, activation of the TRH. We'll see some more on that in a moment. The PIT1 and its splicing variants and the regulation of the TSH beta gene expression. Other transcriptional factors involved in TSH beta gene expression, such as cyclic AMP, steroid hormones, other hormones, neuropeptides, and cytokines. So plenty of reading for you to become a relative expert on the hypothalamo-pituitary thyroid axis. Regulation, physiological regulation in humans, the normal physiology. We don't deal with pathology or pathophysiology in this course, but you might be interested in reading some of that information regardless. Mechanisms involved in hypothalamic-pituitary thyroid suppression in non-thyroidal illnesses. This might be interesting in reading in that it covers the effects of hormones and neuropeptides. So that's very relevant to our approach to the study of neuroendocrinology in this class. And the remaining portion is somewhat clinical, so you shouldn't need to concern yourself with that. And then in addition, on the link within the course materials, the ACNP psychopharmacology, there is the Thyrotropin-Releasing Hormone, focus on the basic neurobiology. So this is a very interesting link. Back to the chapter. The distribution, intraneuronal colocalization, biosynthesis. This figure 2 is very interesting. We'll discuss this in the PowerPoint presentation. So you can see from the Trh gene, get the Trh mRNA with translation, the pre-pro Trh. We'll talk about the processing to the pro-Trh, enzymatic excision, producing this molecule, amidation, and cyclization to end up with the fully biologically active tripeptide molecule. So there's terms that pop out: post-translational processing, then regulation, release, receptors, the anterior pituitary. We won't really focus on central nervous system, but we studied enough so that we should be able to appreciate that. Here's a discussion of the enzymatic inactivation. So you see it's the flow for now the focus on a single particular axis. It's the same flow as what we studied with our general models and with our introduction to these, and now we focus on the detail. The anterior pituitary function, central actions covered again, and then it gets into clinical information that isn't relevant to our objectives in this course. And then a final comment on the synthesis. So those are some of the sources for reading to reinforce what I cover in the literature, in the PowerPoint presentation, at the end of which is a link to the source of the information in this PowerPoint presentation. I in no way pretend that this is my original product. It is a presentation of information that's available freely on the internet. And just let me find it here. Forgot to load it. So now a discussion on the HPT axis. Here again you see my favorite picture of the individual with the serene look on her face and the hypothalamo-pituitary thyroid system being illustrated here, the 3-gland, 3-hormone aspect. And this view, I'm just going to set my pointer as I make my other point here. This image of the individual with that serene look on her face reminds us that there is an emotional component related to the HPT axis. in individuals with hypothyroidism, quite often depression is a major symptom or outcome of a dysfunction of the HPT axis. So there are effects on the nervous system that go beyond metabolism and regulation of the HPT axis, which is, of course, reflected here in this illustration, 3-gland, 3-hormone model system with the thyroid hormones producing negative feedback at the level of the pituitary and the hypothalamus. So let's begin a more detailed study of this axis. Excuse me. You should recognize this figure because we used it as an example when we talked about the synthesis of peptides and their genes. So for the thyrotropin-releasing hormone, again, we start with hormone 1 in gland 1 and work our way through and then ultimately talk about negative feedback or the regulation mechanism. It is the tripeptide, the glycine, histidine, proline. The pre-pro TRH, there are 5 progenitor sequences for TRH, as you recall. The glycine, histidine, proline, excuse me, that's glutamine, isn't it? And glycine at the carboxy terminal end. So the glutamine is a post-translational conversion to polyglutamyl by glutaminyl cyclase. So that's the cyclization that we talked about. The glycine is necessary on the carboxy terminal for amidation of the proline residue in the mature TRH. These post-translational processes not only contribute to the biological activation of the pre-pro TRH, but also to the stabilization so it has a longer half-life. You will find in your readings a discussion of the degradation of TRH and the enzymes that are involved in that, and I urge you to give that some close attention because it's a prime example. Here you have a small hormone tripeptide. We predict it would have a short half-life. What are the factors involved in creating the half-life that it does have? If the half-life is longer than what we expected, what are the characteristic features of this phase of the regulation of this neuroendocrine hormone that extend its half-life? So there are 5 spacer peptides as well as a signal region. Remember, from pre-pro to pro-hormone, the signal region or the signal sequence of the initial transient or nascent peptide within the ER is enzymatically cleaved, and then it is the pro-hormone. So there are 5 spacer peptides as well as the signal region that are generated from the ultimate cleavage of the pre-pro TRH hormone. There are N and C terminal flanking peptides. The N-terminal flanking peptide has 7 non-TRH peptides, possibly produced by the enzymatic cleavage at paired basic residues. So arginine, paired basic residues are prime targets for enzymatic cleavage, such as by the carboxypeptidase B-like endopeptidase. This slide is showing us a view of the TRH gene, and there is a single copy in rats that contains 5 copies of TRH. In humans, the single gene contains 6 copies of the TRH coding sequence. So then here we see at the level of the messenger RNA and the genomic DNA, the representations, classical representations that we've discussed all along. There are 3 exons and 2 introns, exon 1, exon 2, exon 3, and the 2 introns. The exon 1 is the 5' untranslated region. We see that projecting on up to the messenger RNA molecule. Exon 2 is the signal sequence for the N-terminal and contains the N-terminal peptide. So here's the putative signal peptide noted here. And of course the remaining part of it from exon 2 is going to be the N-terminal peptide. So there's a site of cleavage somewhere in here. Exon 3 is the remainder of the pro-TRH molecule. Now remember as well, exon 3 is containing a 3' untranslated region upon which the poly-A tail would be added. So note the scale bars here, 0.1 kilobases for the messenger RNA, 0.2 kilobases for the DNA. Next then, of course, the post-translational processing of pro-TRH. We already talked about the signal sequence being cleaved on the nascent peptide or protein as it is first synthesized and translocated into the endoplasmic reticulum lumen, or the cisterna of the endoplasmic reticulum. Subsequently, the pro-TRH will progress through the ER, the transport vesicles into the Golgi, and even after the Golgi into the immature granule, secretory granules, or the type of vesicles for neurosecretory hormone, secretory neurons, neurohormonal secretory neurons. Whatever. You know what I mean. So the mature TRH is found in the cytoplasmic regions, remote from the Golgi. And remember, the vesicles can accumulate within the soma and even into the dendrites, but also is transported anterogradally down the axon. So somewhere between the immature secretory granules, just fresh from the Golgi, on out into the cytoplasmic within the soma, and on down through the axon, there is the enzymatic cleavage of the pro-TRH, such that mature TRH that can react with the antibodies specific for it now becomes detectable. So the cytoplasmic regions containing the mature secretory granules, therefore, are where we find the mature TRH, especially in the axon and down in the median eminence, as we had mentioned before, at the neurohemal junctions. So you'll even recognize this illustration here of the staining for the pro-TRH using a specific antibody, maybe to one of the terminal peptides that doesn't include a TRH sequence and doesn't cross-react with that. So we see the third ventricle here and the paraventricular nucleus on each side of it. Keep that image in your mind, because we'll see another image in a while looking at the messenger RNA through in situ hybridization in this same area for the pro-TRH. This was that higher magnification showing the localization in the perigolgi region, the immature vesicles. And then, of course, the median eminence where there's no staining, whereas on the right-hand side, panels A, B, and C show different levels of the rat median eminence or stock median eminence, and the pars nervosa of the pituitary gland containing axons immunoreactive for the antibodies specific to mature TRH. You see the capillary loops of the primary portal plexus in the median eminence in this view here, the more rostral median eminence. Then let's begin to ask ourselves about the negative feedback. We saw that illustration on the first slide. Gland 1, hormone 1, gland 2, hormone 2, gland 3, hormone 3, thyroid hormones with the negative feedback. So the thyroid hormone receptor cell in that essence then would have to express the gene for the thyroid receptor, and we'll see that there's free T3 and T4 that can enter the cell as well as diffuse out. Remember that's freely diffusible. And then 5' deiodinase is an enzyme that converts T4 to T3, and then the T3 translocates to the nucleus, binds to the thyroid receptor, targeting the TRE, the thyroid hormone response element on target genes. So this is our basic model, and we'll give more details. Now let's ask the question how negative feedback works by hormones 3 at the level of the hypothalamic paraventricular nucleus, gland 1, hormone 1. First, what we note here in this histological section of a rat brain is the paraventricular nucleus, just the left side, showing this location where some wax, beeswax, was deposited that contained T3. And with a drug, these animals were used to treat these animals. The case of hypothyroidism, or the condition of hypothyroidism, was induced. Therefore, we would expect, with the removal of hormone 3 in those animals, we would expect hormone 1 and hormone 2 gene expression to increase. In the lower panel, we see another section from this animal, or the group of animals treated in this way, where the third ventricle is in the center, and we see the paraventricular nucleus on each side. And of course, the left side was where this wax with T3 was implanted in these hypothyroid animals, and the right side did not have the implant, and we see this extremely high level of signal. This is dark field illumination for gel emulsion covered section that had been hybridized with the probe to the pro-TRH messenger RNA. And so there's a high density of silver grains on the right side, but hardly any on the left side, where the T3 was able to diffuse and have an effect on those TRH neurons. Now let's take a look at the figure on the left. I'll read the figure legend first, and then we'll look at the figure to evaluate these statements. Panel A is the northern blot of pro-TRH mRNA. So this is in situ hybridization. If you homogenize the hypothalamus, run the isolate the messenger RNA, run it on a gel, probe it with a similar type of a probe that you did with the in situ, even radioactive, and expose X-ray film. There's newer methods of detecting it now. But anyway, you get to visualize the bands of the messenger RNA, and you can read the optical densities of those with a computerized system. So that's the northern blot when you use gel electrophoresis to separate and then blot onto a membrane and probe with a cDNA or cRNA probe to image it. That is the northern blot approach. So you're separating messenger RNA. So it's from euthyroid, which is normal, lanes 1 through 6. So normal levels of pro-TRH message in the hypothalamus. And hypothyroid, lanes 7 through 13 rats, as you see here, extracted from the paraventricular nucleus. So a hybridizing band for approximately 1.6 kilobase at the arrowhead is present in the paraventricular nucleus extracts, lanes 1 to 5 and 7 to 12. So we noted that something is lacking in 6 and 13, so what did they put there? But not in the cerebral cortex extracts, lanes 6 and 13. So an internal control, negative control to demonstrate that their signal was specific. The graphic representation is shown in panel A as well. I guess these are all panel B. Both of these graphs are panel B. Graphic representation of the hybridization bands above is analyzed with computer densitometry. A significant increase in the pro-Trh mRNA occurs in hypothyroid animals. This data is shown as after normalization of the RNA samples confirmed by the absence of a significant difference in the ribosomal RNA in the two groups of the extracts. So an internal control also for the to normalize to the amount of messenger RNA that was added to the gel in the northern blot analysis. So let's take a look at this. Here's the arbitrary density units, 10 times 10 to the minus 3, 0 to 160. Th mRNA on the left and 28 sRNA on the right. We can see the striped column is the euthyroid and the shaded column is the hypothyroid, and obviously there's a very significant effect size, the differences between the means. The standard error bars do not overlap. This one above and this one below are far from each other. So obviously a significant difference at the statistically significant difference at the p less than 0.01. No statistically significant difference between the 28 sRNA between the two conditions. In the figure on the bottom left, we can see days on the x-axis and 3 axes otherwise. We have pro-Trh mRNA in the arbitrary density units, TSH in nanograms per mil. This would be plasma TSH, of course, and the hormone 3, T4, in micrograms per deciliter. So obviously, on day 0, these have a normal relationship with each other, and over the days of being treated to be hypothyroid, the pro-Trh mRNA obviously increases significantly with reference to this axis on the left. The TSH, obviously a significant increase, so we would suspect that the Trh peptide levels, synthesis and secretion would have increased significantly similar to the message level. The TSH plasma levels, a typical assessment of thyroid HPT function from the plasma sample, and then look at T4 levels. Boom, dropped way down and hardly detectable. So by day 4, when they were hardly detectable, at baseline, probably at threshold level of detection, close to 0. This is the scale over here. Then we see the corresponding increase in hormones 1 and 2. This slide is taking us now to think about the effect of TRH on the release through exocytosis in the anterior pituitary cells. It also is an example of the complex interactions of the HP axes at the level of the pituitary. And this study from the Journal of Neuroscience, volume 15, issue 7, pages 4982 to 4991 in 1995, demonstrates an effect on the lactotrophs. Lactotrophs obviously synthesizing and secreting prolactin. So let's first take a look at our illustration here. We have 1, 2, 3, 4 graphs that are showing calcium concentration intracellularly in nanomoles. So this is performed through a method of getting a calcium fluor molecule into the cytoplasm of the cells. And when intracellular free calcium increases, it generates light, and then you can generate a standard curve to convert that light emission to nanomolar concentration of the intracellular calcium. And we see in the lower part, and for each of these panels, a block here, of course, that's representing a common treatment to all four of these individual lactotropes. CRH, LHRH, or GnRH as we know to call it now, GHRH, and TRH. So not all the lactotropes respond the same way as far as increased intracellular calcium. So this figure is showing the effects of hypothalamic releasing hormones on intracellular calcium concentrations in 4 representative single cells. The cells were stimulated with solutions containing 4 nanomolar of these molecules, including, and then 100 nanomolar of TRH as shown. And 3 observations were made on the calcium influx. The transient triggering of depolarization, therefore we think of voltage-gated calcium channels in the membrane, and extracellular calcium-independent exocytosis. So whenever we think of depolarization being significant and extracellular calcium-independent exocytosis, we have to ask ourselves what's really going on here and what is the essential source of intracellular calcium. We know that it's from the endoplasmic reticulum, and the IP3 second messenger system is being implicated. So even though there is a depolarization, the intracellular increase in calcium, or the exocytotic events, are independent of the extracellular calcium, and this occurs within a half of a minute. That facilitates the depolarization, evoked exocytosis, while inhibiting the voltage-gated calcium current within 3 minutes. So it's a prolonged effect, making us think definitely metabotropic. Here, let's note the time scale. This is 2 minutes, so some of these events extended for quite significant periods of time. And then there's further enhances the depolarization evoked exocytosis by increasing the high voltage activated HVA calcium channel current, the PKC, protein kinase C. The C reminds us of the G sub PLC for activation of phospholipase C, and the diacylglycerol and IP3 are the products of that. The diacylglycerol activates the PKC. So the PKC activation accounts only for this third effect. I realize that doesn't immediately make a tremendous amount of sense to you. You can, of course, go and download the research article and read it for additional details. I anticipate you have those skills by now. What I wanted to point out with these 3 statements was the importance of beginning to think about the source of calcium that's responsible for the biological effect that is being assessed. Is it all extracellular, or is it all intracellular? Another way of stating that, is it independent of extracellular calcium, or is it independent of intracellular calcium respectively? Or do both contribute? Do they contribute at different times? Sometimes extracellular is early, unlike what we saw in the case of the TRH here. And then the intracellular will follow a bit later. So that's why this study was performed, and it found evidence that basically almost intuitively is the opposite of what you would expect. You'd expect depolarization and opening of voltage-gated calcium channels in the membrane to bring about an influx of extracellular calcium, and that would be responsible for the earliest exocytotic events related to increased intracellular calcium. But indeed, that is not the case here. we typically think of this type of an effect, depolarization, voltage-gated calcium channels open, and you get exocytosis. But we have to remember that the Trh receptor is metabotropic, so all of these effects are mediated through metabotropic mechanisms, so it may be IP3 dependent, as we had seen. Certainly the PKC component of the intracellular signaling pathway for this second messenger system, dependent upon the diacylglycerol activation, is responsible for one part. So it may indeed be that the IP3 was contributing to this very early exocytotic event. And then somehow there's a sustained effect. So now discussion of the Trh receptor. So we see that the Trh, obviously, if it's going to cause exocytosis, remember we link, and this is where your reading will be important, we link release with synthesis. So Trh stimulates both synthesis and release of the TSH. So we need to think of how the signal gets to the nucleus to target the TSH gene. You saw that that information is relevant information, at least, is available on the web links that we pointed out at the beginning of this lecture. We noted that the CRE was one of the sites. However, the previous slide implicated Trh receptor with the IP3 mechanism. So can it be coupled to both the adenylate cyclase, cyclic AMP, and the phospholipase C IP3 mechanisms for, or I guess PKC mechanisms for targeting the nucleus. Anyway, let's move on. Now we think of the receptor itself. So the human Trh receptor spans more than 30 kilobases, 3 exons and 2 introns. So a lot of memorization coming up, as you can see. Intron 1 exists in the 5' untranslated region. This is beginning to sound very common. Intron 2, more than 25 kilobases in length. It interrupts the coding region, the open reading frame between the 3rd intracellular loop and the beginning of the putative 6th transmembrane domain, also found in the chimpanzee and sheep. So remember when we talked about the subfamily of receptors that are G-coupled, that have the 8 transmembrane spanning segments. And now we're at the point of having to memorize which intron contains which portions, intracellular loops, extracellular loops, transmembrane spanning segments. So this is an example of integrating that basic study to the specifics for the Trh receptor. So intron 2 is between the 3rd intracellular loop and the beginning of the putative 6th transmembrane domain. So you should draw out the 8 transmembrane spanning segments and look for the 3rd intracellular loop, and then say, okay, that's the 6th transmembrane domain that would be subsequent to that. Exon 3 encodes the rest of the coding region and the entire 3' untranslated region. So the promoter region does not contain a TATA box or a CAT box in the appropriate location. So this begins to signal that there might be some strange type of alternative splicing that might be going on. A 541 base pair intron 1 is conserved between human and the mouse. So these might not be the most interesting observations, but hopefully you can pick from that information what you think should be the most important parts to become very familiar with for being tested over this material. The human Trh receptor, again, for promoter analysis, it determined that there's a cell type specific reporter activation. So you can read on that in the the link that I showed you. There are 2 research articles from which I gathered this basic information. Sequencing revealed putative cis-acting regulatory elements for several transcription factors. There's a table and figures listing those on the links for your assigned readings. Transfection studies showed a response element for the anterior pituitary specific transcription factor, PIT1. So the cell-specific gene expression reflected here, and tissue-specific gene expression reflected here. Next then, let's think about the target of this receptor, the thyroid-stimulating hormone. Remember that this is in this family of heterodimers that share a common gene product, the alpha subunit, thyroid-stimulating hormone, luteinizing hormone, and follicle-stimulating hormone. And this includes, of course, the placental hormone, the chorionic gonadotropin, that is not shown here. The thyroid-stimulating hormone, also known as thyrotropin, is secreted from cells in the anterior pituitary called the thyrotropes. Finds its receptors on the epithelial cells. Remember, this is not an active finding, but I shouldn't have to repeat that anymore now. In the thyroid gland, it stimulates the gland to synthesize and release thyroid hormones. The TSH is a glycoprotein hormone, so we can already picture several different aspects of post-translational processing. We have glycosylation and complexing with the 2 subunits to generate the heterodimer. So there's the 2 subunits non-covalently bound to one another. The alpha subunit of TSH is present in the 2 other glycoprotein hormones, the FSH and LH. And in primates, the placental hormone, chorionic gonadotropin. Each of these hormones also has a unique beta subunit, which provides receptor specificity. In other words, TSH is composed of alpha subunit bound to the TSH beta subunit, and TSH associates only with its own receptor thereby. The free alpha and beta subunits have essentially no biological activity. So the secretion of the thyroid-releasing hormone, and hence TSH, is inhibited by high blood levels of thyroid hormones in a classical negative feedback loop. The thyroxine inhibits responsiveness of thyrotropes to TRH, makes us think of receptor downregulation, most likely at the level of gene expression. Thyroxine inhibits synthesis of TRH at the hypothalamus. So we predict, and we saw a TRE on the promoter for TRH gene. And the 5' deiodinase converts the T4 to T3 in the target cells for the genomic effects. And so this, the presence of the deiodinase, forgive me, within the targeted cells of the hypothalamus and the pituitary is essential to establish this possibility. As well, although I haven't performed the literature search, I am assuming that the thyroid hormone receptor gene is also expressed in these target cells. Well, now what I'm going to do is just take a moment to assess where we are on the slides. We're on 14 of 33. Let me just talk a bit about the thyroid gland and the effect of TSH there, and we'll split this presentation into 2 lectures thereby. So we see in the left-hand panel an illustration of the parathyroid gland sitting upon the thyroid gland, and this is a histological stain at low magnification and higher magnification showing the follicles with the follicular cells surrounding them. This is the interstitial space, and scattered throughout there will be capillaries most likely in these locations that we see here. Rich capillary bed within any endocrine organ. And we're now beginning to place ourselves on the TSH molecule, imagining ourselves diffusing out of the capillary into the interstitial fluid and bumping into all these cells in here. And when we contact our TSH receptor, we cause a response in the follicular cells of the thyroid gland. And here we see at high magnification the follicular cells. The apex of these cells face the follicle itself that's filled with a colloid protein complex called thyroglobulin. And the base of the cells face the interstitial fluid compartment that is exposed or connected with the space surrounding the capillary beds. So we'll end this by writing our TSH molecule, squeezing between here, binding to our receptor along the way. Some obviously got to these cells. Look at how they produce this scalloped appearance as they have degraded the colloid to synthesize T3 and T4 that then diffuse out and find a capillary and are washed away in the plasma. So I'll end with that slide, and whoops, I'm trying to find the right button here. I'll end with that slide and begin the next lecture on that slide as well, and continue through the remainder of the slides on the hypothalamic pituitary thyroid axis. I appreciate your time and attention, and I'll see you again soon. Or at least you'll see me.